Hey everyone, welcome back. We are on to spermatogenesis and oogenesis. Oogenesis. Difficult word to read. um, so, again, this is a chat GPT rendering of what spermatogenesis is. For some reason, it's also fertilizing. I don't know. Just, like I said, fun. This weird yarn looking aesthetic to it. We're going to talk about the way I kind of decided to do this course as opposed to the textbook Griffith et al. That I had used before is just kind of go in the more in the order of development. So starting with spermatogenesis, so the male and female creating the gametes needed to procreate, and then sex happens, and then we have fertilization, and then the creation of a new individual, right? So we're starting with at the very beginning, spermatogenesis. So we did a little primer on meiosis just to kind of remind you of how that all works compared to mitosis, because it's very important in both of these, in both spermatogenesis and the creation of the egg. So let me get my pointer here. Pointer options. Laser pointer. Okay. So remember, each cell consists of a haploid nucleus, and they also have a propulsion system, right? So we talked about meiosis and how we kind of get to this. Now we're going to talk about how the actual, once we have these haploid cells, what is actually makes them into a sperm cell, right? So it can, each cell consists of a haploid nucleus, right? So we have half the genetic information, which is important. And it also has a propulsion system, right? So these sperm have to travel. They have to get to the egg. It's not random chance, that an egg and a sperm meet up. That one of them has to go to the other. And so it also has a sac of enzymes in it, and these enzymes are important for the sperm to be able to actually penetrate the egg. This is really important in things like sea urchins and things where, like, for instance, we're not going to get too much into non-human, non-mammal stuff, but like sea urchins and fish and stuff that just release their sperm into the water, right? So if you know how fish work, they lay a bed of eggs, the female does, and the male comes by and just releases sperm near them, and then the sperm go and penetrate the eggs and start the process of development. But you have to imagine that if all these fish in the pond or the ocean or whatever are doing this, there's tons of different sperm around, right? So you need very specific chemical cues and signaling, and also these different enzymes that are kind of specific to each species that allow and prevent different species from fertilizing eggs of a different species, right? So you don't want like a sea urchin sperm trying to fertilize a barracuda egg or something like that, right? Anyways, so they have these enzymes inside the sperm. So the centriole here produces a long flagellum, and this is at the posterior end, so that's the tail of the sperm. We have this kind of like tadpole shape, right? The Golgi apparatus up here. So this will eventually form the acrosomal vesicle at the anterior end in the future, and this is what contains, you can see it's kind of forming here in this last picture here in the head, and so this is what contains those enzymes to digest the protein and sugars that are coating the egg and kind of protecting the egg. There are mitochondria down at the bottom here. They collect around the flagellum near the base of the haploid nucleus here, and they are make up kind of the neck of the sperm. Remember, mitochondria are the powerhouse of the cell, so they're near where the tail is in the motor proteins. So the remaining cytoplasm after, so you can see as this is developing, the remaining cytoplasm is jettisoned. and the nucleus is condensed. So we're we're going for speed, baby, right? So these sperm, I'll get into a little talk about Hopi Hoekstra's experiment research up at Harvard, but these sperm are competing to be the first one to the egg, because only one gets to fertilize the egg, and then it's kaput, especially in humans. I mean, maybe we have multiple eggs released, right? That can happen in rare occasions, but you have millions and millions of sperm in one egg, so it's a race. Who gets there quickest is the champion, right? And they get to produce or pass their genetic information down to the next generation. So here's just a kind of a GFP, or a fluorescent protein tagged. This is a bull's sperm. So the DNA is stained blue. This is called DAPI. That stains. Okay, make sure my pointer stains DNA. Mitochondria are stained green here, so it looks like they're GFP tagged. And then red is the flagellum. This is a tubulin stain, so it's kind of a structural protein that kind of makes up, that's within the tail of the sperm. Whoops, did I skip one? Okay, yeah. All right. So let's look at the egg here. So when we do a lot of this developmental biology stuff, a lot of it is done with not humans, right? Because it's harvesting eggs is difficult. B, especially if we get to the point of actual fertilization and things of that nature, you're not going to mess with, aborting development and stuff like that, right? So a lot of it's done with sea urchins, things that usually have larger sperm, they're easier to see, etc. So this is a sea urchin egg, but most of this evo-devo is this idea of evolutionary development, where through the process of evolution, a lot of these reproductive methods and theory and mechanisms are all kind of conserved, right? So this is a sea urchin egg, but like I said, most of this applies very directly to humans. So you can see these little things around are the sperm, right? These, like, you can see the heads, you can kind of very fuzzily see the tails around here. And around here is a jelly coat. And we'll kind of get into that when we talk about the oogenesis part. And we also have this, pronucleus here. In this case, in some species like sea urchins, the nucleus is already haploid, and so there wouldn't be these polar bodies that we'll talk about in a second. Like I said, there's slight differences in these different things. But we also, so you can see the jelly coat, this vitelline envelope, which we'll talk about, exists around it. We'll talk about this when we talk about the actual process of fertilization. We can see the membrane, but really what I want to point out is like the size difference here between the sperm and the egg, at least in sea urchins. So, what does a egg have compared to sperm? Remember, sperm's got the proteins, the enzymes to get into the egg. It's got DNA. It's got some mitochondria. It's got a tail. It ejects jettisons all the extra cytoplasm. It's just going for speed, baby. What does an egg have? So egg has a lot more. That's why it's bigger, right? So nutritive proteins. So you need the energy and amino acids for early development. And so these are like in chickens and stuff. They have these big yolks, right? So that type of developmental stuff. That's actually more because it's not carried within the chicken as it's developing. Anyways, but they also have these proteins for early development, right? Ribosomes and tRNA. So as soon as those genes or those chromosomes come together, you got to start using that information to start building your new person or new embryo. So you need ribosomes and tRNA to actually carry out the creation of these new proteins in this new cell. Messenger RNA, first blueprints of the developing cell. Morphogenic factors, so these are transcription factors and paracrine factors that we'll get into a little bit later. But these are provide positional information, right? So how do we know left from right, up and down, head to tail, etc.? We have these paracrine and juxtacrine factors that are already in the cell at the beginning. And it also has some depending on, so this depends on the species. So protective chemicals. So DNA repair enzymes and antibodies, yes, for humans. UV filters, bad tasting chemicals, that's more for like frog eggs and things that are preyed upon, etc. We don't really need UV filters because you're going to be developing inside a womb, right? But for other species, they've evolved these other things as well. So one thing I want to point out, like a soapbox thing, this let's see, it would, I guess, be ribosome, so ribosomes and tRNA. Really what is in, one thing that is in the cells that makes a movie like Jurassic Park not work. So this is like a bummer, because I like the movie. This movie is like way older than all of you watching this, so hope maybe you've seen it. The original. Now we're not talking Chris Pratt. We're talking way, Jeff Goldblum way back in the day. Anyways, so in the movie they find dinosaur DNA from mosquitoes encased in amber, right? And so they take the dinosaur DNA out of the mosquitoes that have been preserved for all these years, and then they like put it into frog eggs, which frog eggs, first of all, if anything, bird eggs. Birds are technically still dinosaurs. Anyways, so that's already a thing. But you put them into, you put it into these eggs, and they're like, okay, now we can create dinosaurs. The problem is the DNA has to go into an egg. Haploid DNA has to go into an egg that has proteins specifically made for that species. So these transcription factors are going to look for binding sites to start this transcription and translation of proteins, or transcription of mRNA and then translation of proteins. They're going to look for binding sites that they're designed to find. So the odds that if you take dinosaur DNA and you put it into a frog egg or something like even a bird egg, which would be more closely related, the odds of those transcription factors actually matching up with the genome of that species is negative. There's no way. And so that's all because of these transcription factors in the egg are specifically made or specifically have evolved to fit or to match up with their own species, right? Anyways, I'll get off my soapbox. But yeah, so the transcription factors are important. They're there to start the actual process, because you can't just put DNA in there by itself. It has to have something to start using it, right? And those are present in the egg. Okay. So stages of egg maturation at the time of sperm entry. So different species, as the egg's traveling down the fallopian tube, at least in humans, traveling down the fallopian tube, like I said, we're going to be mostly human-centric here because most of you are pre-med. But other species, obviously, sea urchins and fish don't have, they don't have wombs, right? They're just depositing eggs on the ground or on the seafloor. But what I just want to point out is that different stages of the meiotic division for these eggs, different species will fertilize them at different times, if that makes sense, right? So in humans, where are humans on here? Most mammals. So during the second metaphase, so we've already got this polar body being created, and this is when they're finally created, but you can see we already have started. You can see here we have the most mammalian cells are fertilizing. Geez, cannot come up with the word. Different species, though, for example, dogs and foxes, surprisingly, compared to mammals, that's why it's most mammals, not all of them. Really early, primary oocyte. So they have this germinal vesicles before we even have become fully haploid, right? So just to point out that there are still some differences here between mammalian. So you should know that mammalian fertilization happens during second metaphase, and maybe that there are, you don't have to memorize this whole list, there are some differences that like dogs and foxes do it earlier, and different species fertilize at different points in the egg's development. I just wanted to point that out, that I want to give you a little exposure of stuff outside of human stuff. We're going to mostly focus on the human stuff, but just note that there are some differences. Okay. So kind of piggybacking on that last slide here, you can see, I just want to kind of show you this problem, this figure of the release of the egg, right? So we have the ovary here, we have the oocyte grows. It is released from the ovary to travel down the fallopian tube, and it gets fertilized early in its journey, right? So that's why these sperm have to be so mobile, you could say. So, you know, they're entering down the cervix way down here, swimming all the way up and around into the fallopian tube to fertilize this egg, right? And so it's fertilized here, and as it's fertilized, it's still traveling. So we already have mitotic division happening inside the fallopian tube, right? And so it's a blastocyst to the point where it's got, I don't know exactly, I have to look up exactly the cell number that's usually implanted by, but before it implants, there's quite a few cells already created, right? And we'll get into this process of cell division coming up in some future lectures. But yeah, so it's very early here. So again, you see there are these creations of polar bodies. And so the polar bodies, remember, when we are talking of the difference between cells and sperm, or sperm and eggs, the sperm, all we want is, it's just a rocket or DNA strapped to a rocket, right? So all it is we're just going to get my DNA there and get into the egg. That's my only purpose. But for the egg, it's got to be much more nurturing. It's got to carry out the actual, all the information, or all the mechanisms and components needed to grow this new being. And so, like we said, it has all the nutritive proteins and transcription factors and yada, yada, yada. But also, during this meiosis, what it's doing is those polar bodies. So remember, we should have 4 daughter cells, each with each haploid, right? So each have half of the mother's information. What it's doing when it creates these polar bodies is it's, for 3 of those cells, just putting DNA into these 3 smaller polar body cells. And then what it is doing is it's taking all the good nutritive proteins and transcription factors and cytoplasm and everything else that is there to start the growth and hogging that into one cell, right? And so that's why one of the reasons the egg is so big is it's taking all the good stuff except the DNA, because we don't want more than haploid DNA, because if we do, then we're going to end up with doubling our genome each time, right? And it's expensive to keep, and so we're just going to kind of put that in this little trash bin called the polar body, and then take all the good stuff from the end of this meiotic process and sequester it into the egg itself. Okay, so just a quick, we're going to get in pretty soon to the kind of the structure of the egg and the process of fertilization. So if we kind of zoom in on the cell wall, so this is kind of looking at the electron micrograph of the side of the cell here. This is a sea urchin egg again, because we don't mess with human stuff really. So yes, it's a transmission electromicrograph, so we're seeing kind of through it. This shows the microvilli here, these little things. These kind of are help aid in sperm entry in the fertilization process. They look kind of like little docking stations, right, for the sperm head. in the cell membrane. And these are covered by the vitelline envelope, which we'll get into a little bit more during the process and when we talk about the actual fertilization process. So there's also these cortical granules, and these are kind of similar to the acrosomal vesicle for sperm, and they contain these digestive enzymes and mucopolysaccharides, adhesive glycoproteins, and this aids in this is a mechanism that gets released when the egg finally is fertilized, and what it does is it is a way to, we'll get into it more, a way to prevent multiple sperm from fertilizing the same egg. So it's kind of like a protection mechanism that we'll get into during the fertilization thing. But I just want to kind of give you a little idea of what the structure of the egg is. Okay. So this is a mammalian egg right before fertilization. So this is a hamster. And so it is encased in this zona pellucida, which is kind of around it here. And the zona pellucida is surrounded by, so that's this kind of, let me use this over here. I'm using the wrong one. Zona pellucida, and then that's surrounded by the cumulus. And so these are follicular cells, they have follicles, that are kind of nurturing the cell during the release from the ovary. So they're kind of there to like kind of help and nurture and get the cell going, or the egg going, in the process of release. So you can see within the this zona pellucida, which is around here, this is one of the polar bodies, and again, this is what is cast off DNA, right? And so all the cytoplasm and all the good proteins and stuff that are important for the egg have been kind of sequestered or sucked out and put into the main ovum here, and all that's left is the DNA in here. So that's just kind of a trash bin for excess DNA. All right, and lastly here, let's look at, this is just another micrograph. So this is just kind of a zoomed out view of a mammalian egg immediately before fertilization here. So this is kind of, you can see the ovum, and it's surrounded by all these cumulus cells, and you can see that it's kind of a, not only is it a race, but it's almost a maze to get to the egg in this case. And this, what did I say, hamster or mouse for this one? This was a mouse. So there's a lot going on around the egg, generally. So it's not just the egg. Like I said, all this cumulus cells, zona pellucida, we'll get more into that in the next lecture here. But this is kind of just a zoomed out view of the process of sperm creation and egg oogenesis and spermatogenesis, and a little bit of the release and stuff. And we'll get into the fertilization process coming up. So that's it. Like, I like to keep them short. No more hour and 20, 30-minute lectures. I'm going to try to break them into 20-minute chunks. So we'll get into that topic coming up.